Assalamu alaikum and hi guys, this is Bajem here and this is our third video on the third chapter of A-Levels Biology, which is homeostasis. Now this chapter, it's a really simple chapter. It has a really, comprises of really simple topics, simple processes that are not really hard to memorize, so hope you guys understand the video. This chapter consists of these topics you guys can see here: homeostasis in mammals and homeostasis in plants. Homeostasis in mammals. In order to function properly, all of our bodies, we need 37 degrees Celsius temperature because our enzymes in our body that they work optimum at this temperature, and the cell functioning is the best. It's like perfect on it. So our body needs to maintain this temperature. I mean, if it goes down, then the enzymes will start to decrease in activity because the temperature is gonna fall down, and enough activation energy might not be required, might not be given to the reaction. And at high temperature, what happens is that um, at high temperatures, the enzyme they get denatured, and therefore their structures are broken, so that they cannot fit the substrate into their active site anymore. So that way the reactions won't take place. So for this, for these processes to not happen, homeostasis maintains a constant body temperature, which is 37 degrees Celsius. Well, it's not only temperature that homeostasis controls, it's all the body factors that homeostasis controls. Um, there are a lot of these factors. Now I'm going to show you guys these factors here. Now these were the physiological factors that I was talking about. And number one is core body temperature. Remember that I told you guys that the temperature is 37 degrees Celsius and needs to be maintained. Now, number two is metabolic waste. Metabolic waste, for example, our body it has a lot of chemical reactions, and these chemical reactions are called metabolism in general. So, every reaction, has a product, and it has a useful product and it has a excretory product. So this metabolic waste is the excretory product, and these products need to be excreted from our body. It's really important, and it's really crucial to excrete these waste from our body because our body cannot store them, and they can cause a lot of damage to our body if not excreted. Number three, blood pH. Well, pH is really simple and really important that pH stands for power of hydrogen, and therefore, if the pH in our body increases or decreases, it will cause a change in the hydrogen bonding of our enzymes and proteins. Yeah, remember that globular structures in our body have hydrogen bonding, and these globular structures are really essential in our body. I mean, enzymes themselves are really essential. Without enzymes, our body metabolism or our chemical reactions in general won't take place. So the maintaining the blood pH is really essential. Concentration of glucose in the blood. Again, that glucose is really important for us, and if it falls below the baseline, what happens is that we can die by it because our body won't have enough ATPs, and we'll feel like really fatigued, and you know we'll lack glucose. So it's really important to have glucose at a normal level. If glucose level increases in our body, then there will be no place for glucose to be stored, and therefore it will cause a lot of damage. And it's also a reactive substance. So yeah, water potential of the blood. Water potential of the blood also, causes a lot of damage. I mean, it causes the cells to burst. If the water potential around the cells there's a lot of water potential around the cells. What will happen is that the water will enter into the cells, therefore causing the cells to burst because there's a lot of difference in the outside of the cell and inside the cell. So yeah, we learned this before, right? That the cells will burst. We learned this before. That was hemolysis, and remember that was in AS. And this here is concentration of respiratory gases. This is also really important. I mean, of course, exchange of gases in our body, oxygen and carbon dioxide. These both gases, how they're exchanged in our alveoli, and how carbon dioxide is excreted, and what concentration of, I mean, sorry, we shouldn't use the word concentration for gases. What volume of carbon dioxide is excreted, what volume of oxygen is taken in. This is really important also because oxygen is a really active factor in respiration. We learned that oxygen is the final electron acceptor. So if there is no oxygen, if there is less oxygen, it's going to affect our respiratory rates, and it might cause a huge problem if there's less oxygen. Now, this is the important definition that you guys need to learn for homeostasis, which is it is the regulation of internal conditions of a cell or organism to maintain optimum conditions for function in response to internal and external changes. I mean, it's pretty simple, right? This is what I told you that whatever happens outside or inside our body, we need to maintain the internal conditions on an optimum base. They don't have to change with the environment, or the environment shouldn't affect them. Because if the environment affects them, then it's going to be a lot of problem there, right? Because, for example, if there's a lot of cold outside, now if our body gets cold from the inside because of the outside, then all the process will stop. Our brain functioning will also decrease and will be a lot less efficient. So for this to happen, homeostasis is really important for maintaining optimum conditions. Now, there are two types of systems in our body, nervous system and endocrine system. These two types of system are really are the systems that play the major role in the homeostasis of our body. So remember that the nervous system consists of nerves and the central nervous system, which is brain and the spinal cord, and the impulses are transferred electrically. And in our endocrine system, it's a hormonal system, which consists of the ureter, the bladder, the kidneys, the pancreas, and all that hormonal organs. And it travels as chemicals in our body. Now, negative feedback. Remember that negative feedback is a process in which a change in some parameter, for example, blood glucose concentration, brings about processes which return it towards the normal. So if something happens in our body, it returns it to the normal. Of course, remember that we learn in chemistry the reversible reactions. If we add something in the reactants, the products increase. If we decrease something in the reactants, the products also decrease. So this is really important to maintain the both sides of it, right? So same as that in our body, negative feedbacks does the same thing. For example, if our body temperature increases, our negative feedback brings back it to normal. So it does the thing against the change, right? So it has to be equilibrium. Remember this word equilibrium? It has to stay at an equilibrium. So not high, not low. So that's what negative feedback is basically. And in negative feedback loop, there are three processes. A receptor. I mean, sorry, not processes. There are three parts of it. There are three contents of it. One, a receptor or like a sensor, which is really important for something to be detected, right? For a stimuli to be detected. And number two, a coordination system. Then we mentioned these two systems before, the nervous system and the endocrine system. In between where these messages are going to be transferred, these stimuli that is detected, how is it going to reach the final effector? So that's why we need the coordination system and an effector, of course. like muscles, or what's going to the thing that's going to reply back to it. For example, if we feel something hot in our hands, we're going the stimulus is that we know that it's hot, and the thing the sorry, I shouldn't say the thing, the receptor which is detecting it is going to send signals through our nerve impulses. It's going to go to our brain first, then our brain is going to send it to spinal cord, the spinal cord is going to send it to effector that is really hard. So then our hand's going to move away from the hot thing, and so this is the whole negative feedback loop. What happens after negative feedback? Well, what happens is that if there's an increase in factor, the body responds to the factor decrease. I just mentioned this earlier, and yeah, vice versa. This is the negative feedback loop. We don't need to like really learn because I just told you that. Just go through this diagram once. That there's a stimuli, the receptor detects a stimuli, and then this information is sent to the nervous system. Then the nervous system tells the effector to carry out an action. Then the factor decreases, or whatever it was. If it was increasing, then it's decreased. If it's decreasing, it's increased. And then this negative feedback loop happens over and over again. Now this is the production of urea. Remember that urea is a very waste product, and urea is always a waste product in our body, and it is produced in the liver from excess amino acids. Amino acids, remember that amino acids are the building blocks for proteins, and these amino acids they contain nitrogen groups, and they also contain NH3, which is remember that they contain NH3 in them. So yeah. And you know, if we eat more protein, then this protein cannot be broken down. Always remember that proteins can never be broken down. They need to be excreted from our body. So in order to that, in order for that to happen, these amino acids are not always excreted. They're sometimes they're also used as useful energy. But they need to be converted to something. They cannot be directly used as energy. So they need to be converted to a carbohydrate maybe, or maybe another substance. I'm going to show you guys. And to make these proteins available, for example, to release energy from them, you need to remove the amino group. And the amino group is basically the nitrogen containing group. So see here, guys, this is the amino acid, and this is the amino group right here. Wait, yeah, this one is the amino group right here, and this is really important to remove the amino group here. So how to remove? So what happens? I mean, how is this group removed? Well, what happens here is that this NH2 group is just removed, and two hydrogen and water is added, and what happens is a keto acid is formed. Remember that it's an acid because um, there's a carboxyl group here, and there's a double bond O here, so it's called an acid. So always remember that how is it removed? NH3, hydrogen, and uh, water is added. And when this happens, then the amino acid is therefore broken down. Not broken down, we won't say broken down, but we can say it's converted into, or respired into glucose, or not glucose, I mean keto acid. And this keto acid is further, respired or converted into glucose or any other substance that we can directly get energy from. Now, what happens with this amino group? When this amino group is removed, by the way, NH2 is called amino group, and NH3 is called ammonia. So remember that NH2 is called amino group. Now, when this amino group is removed, it's converted into ammonia because this amino group it combines with this hydrogen here, therefore causing it to form ammonia. And ammonia, remember that this ammonia is a really dangerous substance for our body, and it needs to be quickly converted into something, because if it stays for long, it can react, and it's a really reactive substance, and it can, cause a lot of problems. It can cause mutations. It can cause a lot of excretory problems. I mean, uh, when it reacts, it can cause diseases. It can cause, not diseases. I won't say diseases, but I can say like uh, uh, it can cause a lot of shortage. For example, if it combines with red blood cells, it can cause a lot of problems for carrying of oxygen. So it produces a lot of problems. Just know that right now, and therefore it needs to be excreted really quickly. And in order for that process to happen, it's excreted here. Here it's excreted, highly toxic compound and all that, and it interferes with the cell signaling process. For example, here I'm telling you that this is one of the problem. It can decrease the cell metabolism. Another problem. It's alkaline. It can form alkaline ammonium hydroxide, which is also another problem, which disrupts the pH. So how it's excreted? Uh, I'll show you right here. Now, this is avoided by converting ammonia to urea. Urea is another compound that is also toxic, but it's not that toxic as ammonia. I mean, ammonia is really, really toxic, and it it disrupts our body in seconds. If it stays for very long. So urea is less soluble and less toxic than ammonia. And how it's converted into urea is this is how. It combines with carbon dioxide to form urea. Now this is a really essential process here, because why? Because these both are waste products. This is a really good way to, you know, uh, decrease the concentration of waste products in our body and just form more useful products. I mean, water is really useful, so this is a really good process here that helps in the removal of waste products in our body. Now, this is the structure of a human kidney. Remember that there's a cortex here, which is the main membrane, you can say. Not a membrane, I won't say membrane, but it's like a hollow space, right? The main hollow space here is the cortex. Then the medulla is the inner part, these star-like structures you see here. Not a star. It's like a triangular structure you guys can see here. This is a medulla. Then these are these are two. This is a vein. The blue one is a vein, and this one is an artery. Yeah, this is an artery. This is a renal artery, and this is a renal vein. Blue ones is always veins. Remember that they carry deoxygenated blood, and red ones are oxygenated blood. So the cortex here done, the medulla done. This is the capsule. Remember that this outer membrane-like structure you guys can see here. These are capsule. This is like the endothelium you guys can say of um of a kidney. And this is the pelvis here, renal pelvis. You guys can see here this small membranous structure. And this whole tube is the ureter. So yeah. Renal artery carries oxygenated blood. A renal vein carries deoxygenated blood, and the vein, remember that it carries stuff away from kidneys into back to the heart. And arteries, they carry always from the heart to these organs. So they have oxygenated blood to supply to them. And the veins, they have oxygenated blood and deoxygenated blood, which the kidneys have already used, and they're used by the kidneys, and therefore they become deoxygenated. The kidney there itself, it regulates water content, and now this kidney, they have kidney has a functional unit. It has a unit that, for example, everything has a functional unit, right? Um, our muscles, they have muscle cells, which are the muscle cells built up as functional units to form muscles. to form muscle tissues and the muscles themselves. So the same way, our kidney has a functional unit which is nephron. These nephrons are really essential for the important for the formation of urine. Now this is nephron here. You guys can see this one. This is the structure of a nephron. This one also. This is the whole one nephron. So what does the nephron consist of? Remember that this part of the nephron, this one, is in the cortex, and this one is in the medulla. The nephron has a few structures. Just memorize them really quickly. This is the Bowman's capsule. This is a capsule type structure. You guys can see here, right? This is a capsule structure, so this is called the Bowman's capsule. There are a lot of veins, circular veins inside it. So no, I shouldn't say veins. There are a lot of vessels inside it, blood vessels. These small vessels you guys can see, right? These bronze, the vessels. These vessels are called the glomerulus. It's like a globular, like a circular structure, so it's called glomerulus. And outside the glomerulus, these glomerulus are carried inside the Bowman's capsule. So outside the glomerulus is the Bowman's capsule. After the Bowman's capsule comes the proximal convoluted tubule. This is the proximal convoluted tubule. This is the loop of Henle. There are two parts of it. This one is the ascending loop of Henle, and this is the descending loop of Henle, because it goes down from here and it ascends from here. So glomerulus, Bowman's capsule, proximal convoluted tubule. This is the descending loop of Henle, ascending loop of Henle, distal convoluted tubule. So this is another convoluted tubule. First one was the proximal, and the other one is the distal convoluted tubule. And after the distal convoluted tubule comes the collecting duct. Remember that the proximal and the distal convoluted tubule are inside the cortex, and the glomerulus and the Bowman's capsule. They're all inside the cortex. Whereas the loop of Henle, renal pelvis, and the collecting duct are inside the medulla. Now, this is another detailed structure of the same diagram that I showed you guys. But remember that the structure of the Bowman's capsule is not a vessel structure. It's a nephron structure, okay? And nephrons are the functional unit of kidneys. And outside these nephrons, they're surrounded by a lot of blood vessels, and these blood vessels are shown here. Now, these blood vessels that are shown here, the glomerulus I told you guys are blood vessels, and these blood vessels are surrounded in by the Bowman's capsule. So now, where does it start from? It starts from the renal artery. The renal artery takes blood to the Bowman's capsule, and before that, when the renal artery branches in, it's called the afferent arteriole. The afferent arteriole is really thick. I mean, it's a really wide structure, really wide artery. So therefore, uh, the blood pressure is low here because it's wider, so it's low. And here the glomerulus. Now the glomerulus is really small vessels. These vessels are really small structures. So therefore, these vessels are branched and really small, and therefore the pressure is going to be really high here. So remember that the pressure is really high here, and the pressure of blood is really low in the afferent arteriole. After the glomerulus comes the efferent arteriole. This is the afferent, and this is the efferent. Now these afferents are wide, but they're not wider than the afferent arterioles. Just remember this right now, that these efferent arterioles are not wider. They are wide, but not wide enough than the afferent arterioles. Now after the efferent arterioles, it goes into the proximal convoluted tubule, and from the proximal convoluted tubule, we can see that the oxygen is used up, and it goes into the loop of Henle. And after all these processes, it goes into the renal vein, where it becomes, I mean, it's deoxygenated, so of course it's gonna go back to the heart. Um, The capillary, yeah, this is done. Now this here is the formation of urine in our nephrons. Remember that the urine is really important because it needs to be excreted in our body, from our body, because it removes the metabolic waste from our body. This is really important to excrete it. If not excreted, I told you guys that could cause really big problems. So ultrafiltration and selective reabsorption are the two main steps that need to take place for the removal and for the formation of urine. So what happens? Where does ultrafiltration happen, and where does selective reabsorption happen? So the ultrafiltration happens in the Bowman's capsule, which I showed you guys earlier, and selective reabsorption occurs in the proximal convoluted tubule. In the Bowman's capsule, amino acids, glucose, water, urea, inorganic ions, all these, you know, substances that are not really required by our body are absorbed by the what do you call it? Are absorbed by the Bowman's capsule, and their glomerular filtrate is formed. Remember that they are inside our blood vessels. All these substances, they are inside our blood vessels. So they're taken out from the blood vessels into the the Bowman's capsule, and the Bowman capsule is the nephron, part of the nephron. See here, this is the ultrafiltration. Where does that occur? Here. This is the glomerulus. The glomerulus. This this is the Bowman's capsule. Now inside these Bowman's, inside the glomerulus, there are these amino acids and organic, ions and these water and glucose. Now they are filtered out the from the glomerulus into the Bowman's capsule. This is where ultrafiltration occurs. Now here, this is how ultrafiltration basically occurs. Water, blood in the glomerulus is under high pressure. Yeah. So remember that we learned that there's the afferent arteriole here, and this is the efferent arteriole, and this is the glomerulus. So due to the low pressure in afferent arteriole and the really high pressure in glomerulus, what happens is that the substances inside the glomerulus are pumped outside. And remember that the efferent arteriole is also not thick. I told you guys that, not wide. It's not wide enough as the afferent arteriole. So the pressure is really high here. And due to this high pressure inside the glomerulus, these substances, water, salts, glucose, urea, they're pumped out inside the filtrate, glomerulus, inside the Bowman's capsule. And when they're pumped inside the Bowman's capsule, they're called glomerular filtrate. So remember that this is really important, that the pressure in the afferent arteriole is lower, and the pressure in the efferent arteriole is really high. Due to this imbalance in the pressure, there's a very high pressure inside the glomerulus. Due to this high pressure, urea, glucose, salts, and water are pumped out into the Bowman's capsule, and this is the Bowman's capsule right here. Now here in the you guys can see Bowman's capsule. These are podocytes. Podocytes are the substances that make up the nephron, and they're present on the Bowman's capsule, right? You guys can see here, and they're present here. And this is the epithelium of the Bowman's capsule. Now see here, this is the afferent arteriole, which is very wide, and the efferent arteriole is not wide enough as the afferent arteriole. Due to this imbalance here, there's a lot of high pressure inside the glomerulus. And due to this high pressure, these substances are pumped outside. You guys can see these arrows. They're pumped outside, and these pumping outside causes them to go inside the Bowman's capsule. You guys can see these podocytes have gaps between them. This is where they're pumped in from. This is where the minerals they come in from. The gaps between the podocytes. Okay. So due to these gaps between the podocytes, these minerals and all these ions and water and glucose, they're pumped in, therefore to form a glomerular filtrate. Glomerular filtrate because they're pumped from the glomerulus into the Bowman's capsule, so therefore glomerular filtrate forms. This diagram here shows us the movement of water inside the ultrafiltration process. So how this happens is that I told you guys these are the podocytes. These here, the two podocytes are shown, and there's a gap between them. Now these substances, amino acids, and inorganic phosphate and urea, they're pumped due to the imbalance in the pressure in the afferent arteriole and the efferent arteriole into the base, into the, what do you call it, the podocyte. They're pumped into the podocyte, uh, crossing the basement membrane. Remember that there's a basement membrane between the Bowman's capsule and the glomerulus. So due to the it passes through the basement membrane, and this basement membrane does not let everything filter out. It only lets small substances filter out. For example, amino acids, glucose, urea, and inorganic ions, and water. So when these substances, except for water, reach here, it causes the potential gradient of water to decrease inside the Bowman's capsule. And when the potential gradient of water decreases inside the Bowman's capsule, the water from the glomerulus, it also comes here, comes into the Bowman's capsule because there's a high water potential inside the glomerulus. Of course, it's a it's an artery. It's gonna have a lot of water inside it, right? Too, like blood and water. So this water is therefore also pumped from the glomerulus into the Bowman's capsule to maintain a constant water potential. See, you guys can see here lower water potential and regional higher water potential. So therefore, this water is also going to be, it's also going to move due to its pressure gradient here. So yeah. Now this is beginning of the selective reabsorption. This is where the selective reabsorption occurs. You guys can see here. Now in selective reabsorption, remember that active transport always occurs in selective reabsorption, and selective reabsorption occurs in the proximal convoluted tubule. Remember that it occurs in the proximal convoluted tubule and it consists of active transport. Now inside the proximal convoluted tubule, there are a lot of adaptations for this selective reabsorption to occur. And these adaptations are, for example, a lot of microvilli on the luminal membrane. Luminal membrane, remember, is the membrane that is on the lumen of the cell. Lumen, remember, was the hollow space. It's inside it's inside the tube, right? So the membrane that's facing inwards on the tube is the luminal membrane, and these microvilli are present to increase the surface area. Wherever microvilli are present, it's to increase the surface area for absorption. And many co-transporter proteins, you're gonna learn that why are these present, and many mitochondria, I mean plural, so mitochondria, to provide energy because there's a lot of because active transport is occurring here, and remember that active transport always requires energy, and cells that are tightly packed together so that nothing escapes outside or uh, they must pass they must be reabsorbed, and so that they are easily reabsorbed. So yeah. You guys can see here that uh, this is the structure of a proximal convoluted tubule with a vessel. Always remember that it's a vessel, okay, not a vein. I always mistake this with a vein, and I always pronounce it vein. So this is a vessel. Just say vessel. So this is a proximal convoluted tubule. Uh, this one, this is the lumen of the proximal convoluted tubule, the light yellow. These are podocytes. I mean the luminal cells. You can say the endothelium of the membrane, and this is the vessel. This is the basement membrane between the vessel and the proximal convoluted tubule. And you guys can see a lot of mitochondria are present here. And there's also ATPase. I mean, the sodium potassium pump. Sorry, there are a lot of present here too. And there's sodium moving out from here. We're gonna learn about that, but just know right now the structure of it. Now, what is the step number one that's occurring in selective reabsorption here? Step number one that is taking place here is that's the sodium that is present on these on these cells. These are the cells of the proximal convoluted tubule. Now, inside these cells here, there are a lot of sodium ions. Now, these sodium ions, they're pumped by the sodium potassium pump into the blood vessel. Remember that this is an active transport process taking place. Remember that always three sodium ions are pumped and two potassium ions are pumped, and this process always required energy uh, because this was not against the concentration gradient. This is against the concentration gradient always. So remember that this process requires energy, and in order to provide energy, a lot of mitochondria are present here, ATP. So therefore, these sodium ions, they are pumped uh, into the blood vessel. Now, when they're pumped into the blood vessel, the concentration of sodium ions inside the the cells of the proximal convoluted tubule, they decrease, and due to this decrease in the sodium ion concentration, the sodium ion move in from the lumen of this proximal convoluted tubule into these cells. But when they move into the lumen of into the cells of the proximal convoluted tubule from the lumen, they they do this via from by the use of a co-transporter protein. What is a co-transporter protein? It's a protein that always brings more molecules with it, uh, other than the sodium ion. I mean, if there's a sodium ion co-transporter, it's also going to bring another molecule with it. For example, a glucose or an amino acid. So therefore, what is happening here is that the the from the lumen of the proximal convoluted tubule, the glucose, the sodium ion is going to be pumped inside these cells of the proximal convoluted tubule, right? But this is going to happen through a co-transporter, and due to a co-transporter, it's going to bring glucose or amino acid into the cell with them. So it's not going to transport alone, but it's also going to transport with another molecule, which is either going to be a glucose or amino acid. And therefore, what happens is that the transport proteins, the basal membrane, allow the solutes to diffuse down the concentration gradient from the epithelial cells into from like high solute concentration into the blood, which is the low solute concentration. So now, what is happening here is that first of all, sodium from these cells, from the cells of the proximal convoluted tubule, they are being transported by sodium potassium pump into the vessel. When this is done, energy is required. So mitochondria are present, they provide energy. When this is done, the the concentration of the sodium ions, they decrease inside these cells. Due to this decrease of the concentration, more sodium ions they enter the cells. But this does not happen alone. This happens in the process of a co-transporter protein, and due to this co-transporter protein, glucose and amino acid are also therefore carried along with the sodium ion. And when this happens, these transport these there are a lot of transport proteins present on the basal membrane of these vessels. Therefore, so what happens is that these glucose and amino acids, they're therefore transferred into the blood vessel, and this is how uh, they are selectively reabsorbed. Remember that this is called selective reabsorption because only glucose and amino acids are allowed through the co-transporter protein. So they're therefore selected out of the lumen of the proximal tubule. So this is therefore called selective reabsorption of glucose and amino acids. And how is it happening? It's happening through sodium ions. Remember, this is really crucial that sodium ion should be mentioned here. How sodium ion co-transporter transport these glucose and amino acids to it. Now that was ultrafiltration and selective reabsorption. That how those you know, processes occur. After that process, the filtrate, it goes into the collecting duct, and therefore, you know, urine is formed from the collecting duct, and the ureter continues, and it goes into the bladder, and all that stuff. And remember that in the loop of Henle, the water is reabsorbed, and a lot of water is reabsorbed with a lot of minerals, but that is not a part of our syllabus anymore, so we're not going to go to details of that. Now, this is osmoregulation. Osmoregulation is the control of water potential, like the fluids in our body, so it's really important to maintain them, and this is also a part of homeostasis. There are special sensory neurons in our body, known as osmoreceptors, and they monitor the potential of water in our blood. And these osmoreceptors are always found in hypothalamus, and hypothalamus is in the brain. Remember. So remember, if the water potential decreases somewhere in our body, these osmoreceptors they're gonna detect them, and after detecting them, they're gonna send some signals or impulses along the sensory neurons. These sensory neurons are going to take these signals to the posterior pituitary gland. This is another part of the brain. This is like right below the hypothalamus, and these nerve impulses they stimulate the posterior pituitary gland to release antidiuretic hormone. This antidiuretic hormone, remember that there are a lot of questions about this antidiuretic hormone. What happens like about it, and how does it happen, and what does it do? So this is called ADH. I'm just gonna call it ADH from now on. And ADH molecules can enter the blood and travel throughout the body. And ADH causes a kidney to reabsorb more water. This is the role of ADH. ADH causes kidney to reabsorb more water. What happens is that in the loop of Henle, when water is being reabsorbed, right? So water is not being fully reabsorbed. Some of this is also left. But when ADH is released, a lot of that is absorbed. So the urine therefore becomes more concentrated. When there's more water in the urine, it's dilute. When there's less water in the urine, it's concentrated. So there will be a more darker color in the urine. And ADH causes the kidneys to absorb more water, and this reduces the loss of water in the urine. You remember that water is lost in seconds in the urine. So if you want to reabsorb that, ADH is released by our uh, by the pituitary gland. Sorry, by the hypothalamus, and therefore more water can be absorbed in just seconds. And this is how it's done. Hypothalamus detects a change. Therefore, it sends signals to the anterior pituitary gland through sensory neurons. And when this happens, the ADH is released. When ADH is released, the water potential in the where it goes to the kidney, you know, and when it goes to the kidney, where water from the urine is therefore reabsorbed more, and the urine becomes more concentrated. So this was all for today, guys, and I hope you guys understood every single thing. And we covered this chapter of homeostasis in mammals. And thank you so much. And I hope you guys wait till the next video. See you. Bye bye.